Oh gosh, I was trying to avoid this.
Good morning. I am not Jason Chesney. Welcome to the Department of Medicine Ground Rounds. Over the past 2 months, we have been hammered by natural disasters. Houston, Florida, Puerto Rico, Mexico, California. And yet we continue to be reminded that humans can also create disasters as well. Las Vegas, and more recently in New York. Our hearts go out to those people in New York, Argentina, who lost 5 of its members and several others. I have to believe that at some point we will be able to overcome all of these issues in this best way possible. So let me remind you a few people. I recognize that Jason is ready to speak, but a few people that we need to remember. Daniel Rutherford was born this day in 1749. He was a Scottish chemist and photographer, but he was also known for having described what he thought was carbon dioxide. He put a mouse in a chamber and sealed it and allowed the mouse to be there breathing until the mouse died, assuming that there was no more oxygen there. He could still isolate whatever was left by passing it through strong alkali. Okay? It was until Anton Lavoisier came many years later who described that was nitrogen. So he described nitrogen. 100 years later, in 1848, the first totally female medical school was opened in Boston. And it was done by Samuel Gregory, who disapproved of male doctors taking care of childbirth. Okay? So we also remember Crawford Lung. When Ralph Paris and I were at Emory University in Atlanta, we used to go downtown to see patients at Crawford Lung Hospital, not knowing who Crawford Lung was. Well, Crawford Lung was a surgeon who actually was the first who delivered ether during surgery, and that hospital was named in remembrance of him. Yokichi Takamain. Don't ask me to repeat that. Born this day back in 1854, Japanese-American biochemist, industrialist, who isolated adrenaline. How can we not remember Takamain? Okay? And then I'm going to remind you about Severo Ochoa. Most people remember Arthur Coburn. They forget that Severo Ochoa also shared the Nobel Prize for the mechanisms of RNA and DNA synthesis. He described RNA polymerase. But today is about cancer. And we remember E. Kyler Hammond. You know who E. Kyler Hammond was? Okay. You are the Brinkley Chair of Lung Cancer.
Not anymore.
So Edward Kyler Hammond was an American epidemiologist who was the first to link smoking to lung cancer. First to link smoking to lung cancer. In 1957, he was the main research director for the American Cancer Society, and he went to Congress. And he told them about the link. And he told them that the link had been increasing tremendously since evidence emerged in 1915. And he was very concerned that the death rate of lung cancer was increasing in this country. In 1930, he thought it was about 2,500. In 1956, it was about 30,000. Today it's closer to 200,000. Okay? And with that, I want to introduce you our special guest of the day, Jason Chesney, who introduced himself already. But let me tell you a little bit about him. Obtained his MD in anthropology, of all things, at University of Minnesota, and decided to stay there to do his internship and residency, Ph.D. in medicine until 1998. Then he did his internship in New York, and New York Presbyterian, Cornell, who was resident at Memorial Sloan Kettering Cancer Center. He did a clinical fellowship in immunology at Cornell as well. But then he came over to UofL under the tutelage of Donald Miller and became assistant professor here in 2003. And we have observed how Jason has done over the years. Last week, I introduced to you an individual who was nurtured by this institution to become a rising star in the area of infectious diseases. I think the same can be said by our speaker of the day, who was nurtured by this institution to become one of the rising stars in cancer research. And I think we should be proud of that, and he should be too. He's currently professor of medicine, biochemistry, pharmacology. He's a Brinkley Endowed Chair of Lung Cancer Research, and recently became the Director of Medical Oncology, Division Chief, as well as the Director of the James Brown Cancer Center. Well known at the NIH, well funded, holds several patents, close to 100 publications, and I think he's someone who's going to move us or help move us into the next stage of cancer treatment. A guy who has tremendous optimism that someday a cure will be done for that disease. And if it emerges from here, even better. Jason.
Thank you, Jesse. Thank you. Yeah. So I left training and came here, and all of my colleagues back in 2001, when I was making that decision, said I was completely crazy, and they were probably right. But I have really been able to do just about anything in Louisville, and I think this institution is exceptional across from all the other institutions I've been. And I've looked at my colleagues, the co-interns I had when I was at New York Hospital, and some have really risen through the ranks. One became the Hem-Onc chief at Vanderbilt, for example, and then went on to be head of medical oncology at Roche. One's a top leader at MD Anderson. But I feel like I've been able to accomplish more than they could at those bigger name institutions. So I have to say to Jesse and to everyone who's involved in helping the University of Louisville be successful, thank you. Jesse mentioned, I'm going to take a second here, Jesse mentioned that causal link between smoking and lung cancer. It's kind of hard to believe today that was controversial back in the 50s. And in fact, there were several politicians who were having heart attacks, and the thought at the time was that these individuals were having heart attacks because of a high fat intake and not because they smoke like chimneys. And in fact, that causal relationship has now been definitively proven through epidemiology to the extent it can. And we're in the process of a big political move to increase the cigarette tax in Louisville. It's 60 cents today. If we raise it by at least $1, goal would be $1.50. That will translate into about 80,000 youth not smoking, which in turn will translate into a lot of people in 20, 30, 40 years not dying of lung cancer, but also, of course, not dying of cardiovascular disease. So it's a very charged debate right now, but it has to happen, especially in this state, because we have the #1 rate of lung cancer-related deaths in the country. 3,500 citizens of Kentucky die per year. That money that will generate, which is about a third of $1 billion, $350 million per year, will help with the pension crisis. So if you ever get a chance to talk to a state senator or congressman, please mention that. I'm going to talk about the immune system today, and it's interesting. I was in Frankfurt yesterday with Mark Evers, who's the director at Markey Cancer Center, and his focus, he's investing all of his resources in two areas. One is metabolism, because he took all of our metabolism group to UK about 5 years ago now. The other is targeted therapies, and what targeted therapies are is you identify a mutation in the cancer genome, right? And then you use a drug against that particular mutant protein. And great success was CML, chronic myelogenous leukemia. That's imatinib or Gleevec, and now multiple second and third generation agents. That success has not been repeated. Even in melanoma, where there's a BRAF mutation in half of melanoma patients, that really was the second success story. Most of those patients still die of their disease. So that's where he's putting all of his money. He wants to they're going to do sequencing, they're going to identify mutations, they're going to match those mutations with tyrosine kinase inhibitors, and they're going to try to cure cancer that way. We're not doing that. That's something that we do. We do it because it's really palliative, but we're not doing that. We're focusing on the immune system. And I'm going to tell you why we're focusing on the immune system, hopefully in under 45 minutes, so we can have a couple minutes to talk. And this is a video that I think it's very educational for those of you who either didn't take immunology when you were in medical school or took it for 1 to 2 weeks like I did, as well as those who really have learned about immunology but didn't know about the connection with cancer. And Dr. Griffiths did, I think, really a fantastic job with this video. And it's 2 minutes of my talk.
Inside all of us lurks a serial killer. A killer whose primary function is to kill and then kill again. These are cytotoxic T cells, a specialized member of our white blood cells. They patrol our bodies, identifying and destroying virally infected and cancer cells. And they do so with remarkable precision and efficiency. There are about 5 million T cells in a teaspoon of our blood, engaged in the ferocious and unrelenting battle to keep us healthy. These amorphous blobs move around quite rapidly, pushing out their leading edge and probing their environment as they go. When a cytotoxic T cell finds a cancer cell, membrane protrusions rapidly explore the surface of the cell, checking for the telltale signs of cancer. They kill their targets using poisonous proteins visible here in red. These cytotoxic granules move down special pathways in the cell called microtubules to the interface between the T cell and the cancer cell. The T cell punctures the surface of the cancer cell and delivers its deadly cytotoxins. This is very important in our bodies, where cells are packed together, as it focuses the lethal hit on the target and minimizes collateral damage to the neighboring healthy cells. The fate of the cancer cell is sealed. The T cell then moves on, hungry to find another victim.
So I show that because really we learned about that. Most of you here learned about the idea that CD8 positive T cells could kill your own cells. We learned, and I learned when I did my Ph.D. in immunology, I learned that class 1 MHC would present viral antigens, for example, and that CD8 cells that were not clonally deleted would be able to identify these cells. But the idea of tumor immunology, which is over 100 years old today, really became practical and effective only in the last 5 to 10 years. And that's despite thousands of clinical trials that failed, especially in the cancer population. So these CD8 positive T cells that Dr. Griffith was mentioning, what they do basically is they engage the cancer cells and they identify them based on class 1 MHC in association with a small peptide, 10 to 15 residue peptide. And then they release these granules she mentioned, the red dots, that are full of perforins and granzyme B that are essentially like bombs. They blow holes in the plasma membrane. And then the cancer cell, or if you get rhinovirus, the virally infected cell, dies. It undergoes apoptosis. Sometimes it's necrosis, but it dies. And we've known about this for years, for decades, for over a century in some cases. If you look at some of the early literature in the early 19th century, but it never worked in cancer. We couldn't figure out how to do it. And it wasn't until, I would say now 10 years ago, that by accident, a protein was discovered, and the one that's really having a huge impact, and that protein is called PD-1, and that stands for programmed cell death 1. And it was a protein, it was a gene that was discovered in a screen where the investigators were looking for genes and proteins, of course, that were involved in programmed cell death. So what they did was they took a leukemia line, happened to be a T-cell leukemia line, and they induced apoptosis, cell death, and they did knockouts of all the genes we have. It was a shotgun approach. And the first one they got a hit on that prevented the programmed cell death was PD-1. And long story short, this protein PD-1 was knocked out in animals, in mice, and they developed autoimmunities, and that led investigators to say, hey, maybe we can make an antibody against this, and that will lead to hyperactivation of the immune system, which in turn led to tumor experiments. Well, that was completely by accident. CTLA-4 is a little bit earlier than that. That's cytotoxic T lymphocyte antigen 4, and that was the fourth one in a similar series. These investigators, this was before PCR was discovered in the 80s, were looking for surface antigens on T cells, in particular cytotoxic T lymphocytes, that they knew could cause death of cancer cells. And one of them turned out to be CTLA-4, and again, by accident, they discovered that if they inhibited this, that in turn would lead to hyperactivation of the immune system. So these two proteins, PD-1 and CTLA-4, as a class, are called immune checkpoint proteins. And they are surface proteins on T cells that transduce signals to turn them off. And they have ligands, of course, and there's a distinguishing point about the ligands. PD-1, its ligand is called PD-L1 now, and that's a great name, right? It's PD ligand 1. It's easy to remember. what's unique about it is that it's not only expressed by the cells that activate T cells as well as turn them off, but it's also expressed by the cancer cells. And that was really unexpected. And in fact, I think Getz is here, but non-small cell lung cancers, I don't know what the percentage is, 30, 40% of non-small cell lung cancers express PD-L1. So here I am doing all these clinical trials, trying to activate the T cells, and the non-small cell lung cancer is expressing basically something that turns off the T cells. So it doesn't matter what I do, because I don't know about this, because it was only discovered in the last 5 or 10 years that this thing even existed. And it turns out with lung cancer that the higher the expression, the more likely you're going to respond. CTLA-4, on the other hand, its ligand is B7-1. It's only expressed on antigen-presenting cells. It's not expressed by the cancer cells, so it's really an immune modulatory agent or signal transduction pathway, and it's very distinct. So this is what forms the center of the entire talk. So does anybody have any questions before I move on? Because this is the basic mechanism about everything. Because I'm going to focus really on clinical trial results after this. So, we've got a way, now we know that T cells turn themselves off, right? They've got a protein that turns themselves off. And what I believe is that you want to be able to turn off your immune system because you don't want to turn into a big lymphoma, right? You get a cut, you get an infection, you got to activate the immune system, you got to clear the infection, and then you got to turn it all off. You got to turn the immune system off. And this is the mechanism whereby that happens. It's one of several, I should say. And it's absolutely essential. So if we give antibodies that block this interaction between the brake, the PD-1 and the CTLA-4, and the ligand, right, the PD-L1 and the B7-1, then what do you think happens to the T cell activation? Does it go up or down? Come on. It goes up, right? So yeah, it goes up. I know it's early, but it goes up. So if you block the thing that turns the T cell off, it's going to get hyperactivated in certain situations. And so it all started with mouse monoclonals, and then those monoclonals became humanized, which basically means that when you give the antibody, it's just like any other antibody in your circulation. You don't get a reaction to it. And the first in class is a drug called Ipilimumab, and that's an anti-CTLA-4, and that hit the scene in the early 2000s. It was developed by a company called Medarex, and Jim Allison was at the forefront of that, and he's at MD Anderson, and one of the two co-interns I mentioned is now married to him, as a matter of fact. And that drug was a failure. Everybody thought it wasn't going to work, and the early clinical trials thought it wasn't going to work. It actually, the humanized version of it got out around 2002, 2003, but it didn't get FDA approved for a decade. So I got here in 2002, and I had done my Ph.D. in immunology, and Dr. Roman mentioned I did my fellowship in immunology, in clinical immunology, and I heard about this, and I thought, and I was going to the melanoma clinic and realized that everybody died. 95% of my patients died at that time. And, it was kind of a defining moment that, we need to, if you've got a patient population where they all die, they all should go on clinical trials. That was my opinion at the time, and so that's what I did. And I sought out trials that I thought made a lot of sense. And this was one of my first ones, and it was to use the humanized monoclonal against CTLA-4 in these patients. And unfortunately, what happened was the response rate was similar to dacarbazine. That's an alkylating agent that's been used for 4 decades, and not very effective. Doesn't improve survival, but it was only, the response rate was 11%, and everybody thought it was just yet another failure in immunotherapy. But what we discovered took about 3 years. I think we started the trial in 2005. We published it in 2010, but after about 3 years, we discovered that our historical controls were 5% survival, but for some reason these patients were up at 20% survival. So we were getting a 10% response rate. 10% of the patients had their tumor shrink, most of which weren't complete responses, shrink partially, but we had 20% of patients surviving. And this data is now out to 10 years, and it's real data. So that was a big surprise, and that ultimately led 10 years later, from when this whole thing started, to the FDA approving the drug in 2011. It was the first immune checkpoint inhibitor to be approved. And fairly rudimentary immunological assays, but this was just looking at absolute lymphocyte counts. No flow cytometry here, I'm afraid. But this was a dose escalation study that basically defined what dose we were going to use in the phase 3 trials. And as we increased the dose of the antibody that blocked the brakes on the CD8 positive T cells, we increased the expansion of those T cells over time. So we saw more absolute numbers of those cells, nothing antigen specific here. And so this data matured. We figured out, oh, there might be something here. Everybody got involved with trying to figure out how to make this better. We wanted 100% of our patients to survive, not 20%. And that led to combination studies where we would hit both the brakes. Pretty simple, right? You hit the CTLA-4 brake, you hit the PD-1 brake, and see what happens. And this is a colon cancer model. It's an odd choice, but it's a syngeneic model, so that it's an immunocompetent animal. And it is MSI positive, it's CT26, and you get a great response when you hit both the brakes at the same time, which was very encouraging. And so we did a phase 2 trial. This is the first time this was attempted. Obviously, it was a phase 1/2, but where we combined a humanized antibody against CTLA-4 with a humanized antibody against PD-1. In this case, it was nivolumab for the PD-1 and, of course, Ipilimumab for the CTLA-4. And what you're looking at is, you see this is a waterfall plot, and when it goes down, that's good, right? So that's the percentage of baseline for the patients. So we had a bunch of patients have complete responses, and the majority of patients, the vast majority of patients, responded to this drug. This on the right is the Ipilimumab only side, and I mentioned 11%. I think we actually did a little bit better than that, but the cutoff, of course, is 30% with the criteria we use for a partial response. But we saw bottom line is we saw a marked increase when we hit both of the brakes at the same time. And importantly, when we published this, which I think was 15, excuse me, the responses were very durable. This is called a swimmer's plot, and all little arrows mean that they were ongoing at the time of publication. So we were deactivating the brakes on the T cells with antibodies, and the patient's tumors were shrinking, and they were continuing to shrink. It wasn't going away. And I mentioned my 5% survival, and then I got to Ipilimumab, and I was around 20% survival. And now when we did this combination, we were looking pretty good at 60% survival, which was nothing short of spectacular at the time, because we basically operated a palliative care clinic in the melanoma clinic at the University of Louisville. Most of the patients who had stage 4 disease die from the time we saw them within a year. And I love showing this because it reminds me of something I like to talk about, which is that we're going to put surgeons out of business. That's our goal. Right. I don't see Dr. McMaster's here, I hope. But we are. We're going to put the surgeons out of business. So you got a big chest wall mass, right? And then you get your anti-CTLA-4, anti-PD-1, and the mass goes away. And the question is, should we have cut that thing out? It was the only mass this patient had. And, I would argue no, because we could have cut that thing out, but the patient would have had the cancer come right back. And Dr. McMaster knows that. And he sends us patients all the time. So if you're interested in cancer, this might not be a good time to go into surgery. So the bad news, this is the bad news. So the bad news is that with this combination, we are really activating the immune system. And in that clinical trial, we were the second highest accruing site. Sloan Kettering beat us out by a few patients. In that trial, something like a half of my patients ended up in the hospital from side effects. And they're serious side effects. And this doesn't, we probably should have called it what it was, but diarrhea, well, that was all autoimmune colitis. And, here you've got grade 3 or 4, right? So you get hospitalized, that's a grade 3 right off the bat. 54% of our patients had grade 3 serious adverse events. It's kind of a big deal. And of them, we ended up with autoimmune colitis in probably 30%, 11 plus 17. Autoimmune dermatitis, which can be handled with topical triamcinolone. Colitis, we deal with that with a mg per kg of prednisone, and then escalate to dexamethasone, and then we have infliximab and anti-TNF alpha, and then it can go on to mycophenolate and other drugs. We do have a small number of patients. I see Dr. McLean in the audience. We do have a small number of patients who end up with autoimmune hepatitis, which can be, we've had a patient die of autoimmune hepatitis at this institution. He wasn't physically here, but he was one of our patients. And so that's a pretty serious side effect. And in fact, with just Yervoy or Ipilimumab, the anti-CTLA-4 alone, about 1% of patients will die from the treatment. So it's pretty serious business. When you add the nivolumab, it gets that much worse. It almost doubles the serious adverse events. So that's the bad news. This is educational, but it's like it's all happening again for me. So all those patients with colitis were going to ERs. They were being put on antibiotics. They had bloody loose stools, and then they were dying because they weren't coming to us and where we would put them on steroids. Well, what's happening now is with the PD-1 inhibitors, we're getting a bunch of patients with pneumonitis. Anyone who's had any radiation to the lung, like a breast cancer patient from 15 years ago who had some radiation to the axilla, they're ending up with pneumonitis. There's a certain incidence without radiation, and they're being diagnosed with pneumonia and being put on antibiotics. And so, this happens to me all the time, and I say, well, stop the antibiotics and let's put you on steroids. So we put them on steroids, and they do great, and everything's okay. But just that, if anybody's on an immune checkpoint inhibitor that you encounter, whether you're an ER doctor, a surgeon, a radiologist, whatever, think about this. And the immune checkpoint inhibitors are Ipilimumab, nivolumab, pembrolizumab are the most common ones, those three. I propose putting metal alert bracelets on these patients, but it hasn't flown yet. And what's weird about it is you get treated here, right? But then you get your side effects out here. So it really throws the medical establishment for a loop. We don't understand that it's not chemotherapy. These are humanized antibodies that have half-lives of 26 to 30 days. They're in your circulation for a half a year. They're detectable for a half a year. And these side effects don't have to happen right up front. They can happen, this is for that combination, but this can happen a year and a half into therapy. You can end up with one of these autoimmune side effects. And we don't know why that happens. So it's very important to recognize the potential for these agents to cause autoimmunities that are lethal. These people die of them. I forgot to mention, I mentioned dermatitis and colitis and autoimmune hepatitis. Hypophysitis happens, and I see Steve here, and it does happen. It happens. Any patient who walks in with these therapies who's walking around like this, and it's either going to be autoimmune thyroiditis or it's going to be autoimmune hypophysitis. And you check an ACTH for the hypophysitis, you check a TSH, and you can pretty much solve the problem immediately, with some Synthroid or for low dose steroids. The good news, however, is that we just did a follow-up paper not that long ago, I can't remember when, last year, where we looked to see what happened. Because, you give chemo and the tumors shrink, and you stop the chemo and the tumors grow, and then the patient dies, right? That's chemo. You give immunotherapy, the tumors shrink, and they stay shrunk. And this was a 2-year follow-up we did, and it looked virtually identical to the initial objective response rate, as you can see. And then the survival looked, I don't know, and I have to admit this was the BRAF wild type. And the reason I show this is that if you have a mutation for BRAF, that will perturb your data because you can go on a BRAF inhibitor and that'll keep you alive for an extra year. So these patients didn't have that option, and it looked even better than the total population for some reason. But you can see 70% of the patients were alive at 2 years, and it was around 20% 5 to 6 years ago who would be alive, and at 3 years it's about 5%. And so why is this important in the grand scheme of things? Well, it's important because melanoma is by far the most immunogenic cancer. It has the most mutations, by far. And that's shown nicely here with the somatic mutation frequency here. This is the number of mutations that melanoma has. And that's why that's why, frankly, I started working on melanoma when I got here, because I knew that, and that's what I wanted to do. But what's important to note is that, and it is a logarithmic scale here, but there's a lot of other cancers that have a lot of somatic mutations. Melanoma is about 10,000 different mutations have been, in a typical melanoma. But it's not that big of a drop to get to a much more common cancer, non-small cell lung cancer, squamous, and then adeno. And so, the big question is that going to translate into, are these therapies going to work? And short story, but these are all the drugs that are now approved, FDA approved, for all against immunocheckpoint inhibitors. And the top two up there are nivolumab and pembrolizumab. Those are against PD-1. And you can see all the indications. Maybe you can't, but I'll read them. Melanoma, lung cancer, renal cell carcinoma, Hodgkin's lymphoma, head and neck squamous cell carcinoma, urethral carcinoma, MSI, microsatellite instability positive solid tumors. I'm going to stop there because if there's an oncologist in the audience and you've got a patient you're sending to hospice, check them for MSI, because if they're MSI positive, they might respond to an anti-PD-1. The response rates are like 70%. It's remarkable. There are now a third class of agents. They're down here. They're against the foot, PD-L1. And one of them in particular has been FDA approved for Merkel cell carcinoma right here. Pembrolizumab works great with it too. I don't think it's been approved for that. But this all started in melanoma, and now it's spreading across multiple indications. And the goal is to try to figure out ways to improve the efficacy in those indications and then get it to work in indications that it's not approved for, like breast cancer, for example. And so that's what we're doing here. And this is a neat phase 2 trial. It's the same agents that we used in the melanoma way back when for non-small cell lung cancer. And this is just an example of a patient who's very large adeno shrunk significantly after 6 months of a combination of nivolumab and Ipilimumab. And so Dr. Kloker is planning to initiate a phase 3 clinical trial. This is the phase 2 trial, and I ran this one, but he's going to do the phase 3 here because lung cancer is so devastating to the state. So why doesn't it work in everybody? I mean, what's going on here? And I have to admit, I haven't gone into the literature and read all the minutia about what people think. But this is what I think, and it's kind of old school stuff, but it's a combination of, a third of all melanomas don't express class 1 MHC. You can't get a CD8 positive T cell to kill your melanoma cell if you don't have class 1 MHC. So that makes complete sense. We know that there's sort of dysfunctional antigen presentation that happens. We don't know exactly why. Probably the cancer cells are making things like TGF-beta to turn off antigen-presenting cells. We know that when you look at a tumor, it's full of immune cells that turn off the immune system. They're called immune suppressor cells. So when you get a cut, there's an infiltration of suppressor cells so that you don't end up with an overwhelming inflammatory event. Same thing happens in tumors. And then there's a kind of a collective term called T cell anergy. It basically means T cell's there, it's cocked and loaded, it's got the right receptor, it's ready to go, but it doesn't go. And the easy explanation for that is PD-1 expression, but a lot of these T cells don't express PD-1, and they still don't go, and we don't know why. I'm just going to mention this. I couldn't show you the data. It's right now under review, and I didn't get a chance to talk to the communicating author. I didn't want to show the data, but it's really interesting data, so I'm going to say it without showing it, which is that for some reason, so we looked at about 1,000 patients who got PD-1 and CTLA-4, right, over the past 10 years. And there's a strict correlation for CTLA-4 responses. So in other words, with class 1 MHC. So if you have class 1 MHC, you respond to CTLA-4, you're more likely to. If you don't respond. So that fits the dogma. That fits what I learned when I was in school, that if you don't have class 1 MHC, you're not going to respond. The PD-1s, there was no association, which suggests to me that there's some other mechanism in play that we don't understand yet. So for some reason, PD-1s don't require, it doesn't matter if you're expressing class 1 MHC or you're not. So that I just think that's interesting and worth noting without showing, unfortunately, the data. Poor antigen presentation, we're focused on this. We want to improve antigen presentation. We want to improve, we want to reduce T cell energy as well. And one of the approaches that we've taken is we've worked with this guy, Rob Coffin. Rob's a scientist from London. He developed he worked on oncolytic viruses, and about 15 years ago he started this, and about 10 years ago he developed an oncolytic virus that specifically would enhance antigen presentation. And it does that predominantly by having the open reading frame of granulocyte macrophage colony stimulating factor. So it's a gene that expresses a protein that causes the differentiation of antigen-presenting cells that activate CD8 positive T cells. So the whole idea is you activate the CD8 positive T cells and hope that through antigen presentation that you're going to end up with more CD8 positive T cells and an immune response. And this led to a clinical trial that I'm going to show you the results of, but it's a pretty simple strategy. You take a virus, makes GM-CSF, you inject it into the tumor, the virus takes over the ribosomes of the tumor, the melanoma cells, makes GM-CSF, the GM-CSF then stimulates the monocytes to become dendritic cells, they get activated, take up the melanoma antigens, and prime CD8 positive T cells. And the way we did this was we injected the viral agent, it's a herpes simplex virus 1, into lesions that were accessible, It could be lymph nodes. We use ultrasound, et cetera. And then the whole idea was we would get systemic immunity. We would activate the CD8 positive T cells, and those in turn would reduce tumors somewhere else. And the way the trial was designed, we actually saw that. We had significant response rates with in the M1C population, which involves visceral disease. But what was more, so all the, again, waterfall plot, you see all the tumors shrinking there. It was randomized to GM-CSF alone, and you see that was a big increase. But the most impressive thing, this is the durability of the responses with GM-CSF, and this is the durability with the virus. And what this indicated at the time, anyway, which was a few years back, but was that it was a durable response. We had a durable response. We were causing an immune-mediated response and this is a lady from Ashland, Kentucky, who came here and got treated after she was basically told there's nothing else we can do. She'd gotten isolated lymph perfusion. This is back when everybody died. The response rates were really good in the population that had the injectable disease. You know, this is going to be your lymphatic disease, and this is going to be subcutaneous tumors. You know, roughly a third of patients. This is really good for back, 10 years ago. That was nothing short of remarkable, and those were complete responses. We were getting rid of tumors in a third of our patients. So we were really happy about that, and this was all happening, and then the UofL trial was happening, and which is anti-CTLA-4, and the idea was let's present the antigen to the CD8 positive T cells, get them activated, and then block the off switch and allow them to become hyperactivated. So we combine an immune checkpoint inhibitor that will allow the T cells to go crazy with antigen-presenting stimulator, which is a virus that stimulates the immune system to present the antigen. It was a rational approach. There was data in animal studies. The hope was that we would get synergy. I don't know if we reached that, but we certainly had some improvements. And so this was the phase 2 trial. It's the first trial ever to combine an oncolytic virus with an immune checkpoint inhibitor. It was simply put, we injected the virus every couple weeks, and we gave the immune checkpoint inhibitor, which at the time was the only one available, which was Ipilimumab, every 3 weeks times 4, and we looked to see what happened, basically. And this was one of the early patients, and he's still here. This trial opened, 6 years ago, and he's been on in various media, so I'm allowed to show his face and talk about him, but his name's Clyde Sally, and he had these horns growing out of his head. He's very religious, and he would go to church, and they would talk to him as if he were the devil, and he used to joke about that. But there you can see that one lesion on the right side of his head there, and here's CT imaging just to quantitate the size of the lesion. And this is basically what happens. You inject it with the Ipilimumab, and all these tumors that were unresectable, basically, he had multiple prior resections, are gone, and he's alive today. And this was 6 years ago. I think he's on the anti-PD-1 maintenance therapy right now. This is Rob, who came to visit me, who invented the virus with the patient. And I think this is a very special picture, and I like to show it. Our hypothesis was that it would affect visceral disease. It wouldn't just be the ones that we were injecting, but also the stuff elsewhere. And I think we showed that in this trial. I'm going to show you data to prove that. But in this case, this was a right upper extremity lesion being injected and did a great job. It got rid of it completely. The patient actually progressed after 3 months, which we see a lot because that's immune infiltration. And you can see a nice lung lesion in the, right middle or upper lobe there, and then a bunch of what appeared to be new liver metastases, or at least some heterogeneity. I think the radiologists call it new liver mets. But then the lung lesion started to resolve, and it went away completely, and the liver completely cleared, and the patient ultimately had a CR. And so that was very encouraging. Now, was that just from the UofL? We didn't know. But I'm going to show you some data that supports that it wasn't. And so we just published this a couple weeks ago, early October, and what happened was with UofL alone, we did pretty well, which probably suggests that we had an earlier stage population. The less bulk of your disease, the more likely you are to respond to immunotherapies, and that's been shown time and time again. So we had an 18% response rate with an anti-CTLA-4, which was, that we started with that in 2002. That's old stuff. When we added the TVEC, we got up to a 39% response rate. So we roughly doubled the response rate, which was that felt good. And I thought, well, okay, maybe I don't know if we got synergy here, but we at least doubled the response rate with this combination. And this is just waterfall plots showing what happened. And I drew these red boxes around the plots because it really shows it better. But basically, they're the same size. So this area here are the patients who didn't benefit if they got Yervoy or Ipilimumab, the anti-CTLA-4. And that area is the patients when you added TVEC. And so what we saw was the tumor shrunk more, right? That was big. And the regressions were, and there were more of the regressions, the tumor regressions. But this was the most important data in my mind from the paper, which was that our non-injected visceral disease response rate was much higher in the combination arm, which has been a big controversy with this, because you can inject hydrochloric acid into a tumor and it'll shrink, right? You can inject just about anything into a tumor and it'll shrink. And so the idea that we can induce systemic immunity is why we're doing this and why we're working so hard on this. And what you're looking at is we gave the virus with the immune checkpoint inhibitor on the left and just the immune checkpoint inhibitor on the right. And this is about what you would expect. These are just visceral lesions. And what we saw is when we added the virus, we had a marked increase. It's a small population, I should say, 31 and 22. But it was clear that there were more patients who responded viscerally when we added the virus, which would indicate to me that there's an induction of systemic immunity happening from a clinical perspective. And so now we're doing an anti-PD-1 with TVEC. And why are we doing that? Instead, why wouldn't you do the triple therapy, right? It's because of all the toxicity. The anti-CTLA-4 with anti-PD-1, you got a death rate, you got patients going to the hospital, so we're trying the anti-PD-1. And we won't know the results for a year and a half, but we are the University of Louisville is the top site in the world. There's 113 sites going all the way to Australia. We're locked at the top site. We had to ask for more slots in order to accrue, so we're half the accruals in the United States right now. It's going to be a positive study, and I'm going to say that. I know this is being videoed, and I'm not supposed to say too much, and this is an example of a patient on the study, and I can show this because he just got filmed by PBS last week. His name's David Cornelius, and he had a big tumor. And historically, we would send that patient to a surgeon, and it would be cut out. Instead, he went on this trial, and you can see what happened to his left axillary mass. Well, 51 patients enrolled, 6 patients came off, 4 died. One was a heart attack, 84-year-old farmer. One was a drug overdose, 32-year-old guy who had abused drugs in the past. 2 were from progression. The 5th one was gastric adenocarcinoma, another malignancy, and she died of it. And the 6th one responded to adding TVEC, because it's a randomized trial where half are getting placebo, responded to getting TVEC after he came off the trial. So what I just said was about 90% of the patients haven't come off study. 90% of the patients are doing well. And Kelly McMaster's sending patients to us all the time, and he said, I've sent you like 7 or 8 patients for this study, and with the expectation they will come back so I could do the surgery after 3 months when it didn't work, and none have come back to me. What's going on? And I said, well, none have died, Kelly, and they've all responded, which is the truth. So we're really getting to the point where surgery isn't going to have, for metastatic disease, second-line therapy, it's not going to have much role in melanoma, which is nothing short of dramatic. So melanoma is it's 10,000 deaths out of 600,000 deaths in the United States. How's this going to impact the cancer death rate in the country? Well, we decided that we were going to try this in liver metastases, and we opened up the trial here, a phase 1 trial where we injected the TVEC directly into liver metastases, and we did multiple common solid tumor types, including breast cancer, lung cancer, gastric, et cetera. And the first patient went on last year. I think it was in January of 2016, and she had a bunch of liver mets. It's actually kind of hard to see, but that's a breast adenocarcinoma. She had progressed through anti-estrogen therapy, and she started getting, you can see the interventional radiologist, I think that was Doug Caldwell doing that one. It's him and Missy Potts. And she actually responded initially and went on the treatment for 9 months, which was the max for the phase 1 trial, and then she had to come off. And they just did a little story about her. That her name's Janice Adams, so she's out in the public domain. That's her right there. And unfortunately, after 9 months, she started to progress. After we stopped it, we put her on anti-PD-1, and she did extremely well for a year plus, and then died of something completely unrelated and unexpected. although who knows, you can never tell, but did extremely well. And our whole goal with this is to do these injections for these solid tumor types in combination with the immune checkpoint inhibitors so that we can translate the success we've seen with melanoma into multiple solid tumor types. And so I'm going to jump real quickly into the third area that causes resistance, immune suppressor cells. And, you know, immune suppressor cells, you've probably not heard a lot about them. When I went to school, they didn't teach us about them, but the big one is myeloid-derived suppressor cells. It's a myeloid-derived suppressor cell. It turns off CD8-positive T cells in an antigen-specific way. It turns off CD4-positive T cells in both an antigen-specific and a non-specific fashion. And those cells are stimulated by a variety of other cells. And the big one is this cell here called a Th17 cell up there. And Th17s, they're defined by their ability to make IL-17. And so why am I interested in this? Well, those cells probably are part of the reason why not all the patients are responding. And it turns out those Th17 cells are regulated, their programming and their differentiation are regulated by a transcription factor called hypoxia-inducible factor 1 alpha, which was Greg Semens has been studying for 20 years and is important in cancer cells for them to respond to hypoxic environments, and they upregulate glycolysis and erythropoietin and all this other stuff. But it turns out these cells, for some reason, require them. That was discovered predominantly by Eric Dang, who's Chi Dang's son. And we happen to work on a protein that is regulated by HIF-1 alpha, and it's actually probably the most potently induced protein of HIF-1 alpha, and it's called 6-phosphofructose-2 kinase isosome 3, and it regulates glycolysis. That's all you need to know. It turns on glucose metabolism. And we made a drug against it because we were trying to target cancer cells, and the drug's pretty specific, and we got into a phase 1 clinical trial, and along the way we started thinking about this and wondering, we were seeing a lot of weird results, like the drug dose wasn't, you'd require 5 micromolar to kill a cancer cell in vitro, and you put it into an animal, and it would require like 100 nanomolar. There was this like disconnect between what we were studying in vitro and what we saw in whole animals. And so we came up with this hypothesis that if we inhibited this enzyme that was clearly involved in these suppressor cells, we might be able to stimulate immunity. That was the idea. And we did it in a B16 model. B16 is a melanoma model. It's a syngeneic model. And we gave the animals who were bearing these tumors PFK158, which inhibits this glucose regulator. And when we did that, we were able to completely deplete the Th17 cells, and that's shown up at the top there, as well as the myeloid-derived suppressor cells that they promote. And in turn, that led to an induction of effector CD8-positive T cells as indicated by their interferon expression. And so we came up with this sort of model of how this all might be working, where we could hit these suppressor cells. Because right now, there's no FDA-approved drugs that hit the suppressor cells. So it's a really hot area right now. And so this went into a clinical trial, and we did, I can't remember how many, 18 patients got treated, and we had 6 experience some clinical benefit. These were patients, multiple cancer types, who had been treated with everything, immunotherapies, chemotherapies, targeted agents. This was their last leg, early phase. We opened it up at a bunch of institutions, including here, but Lombardi and MD Anderson, et cetera. And this was an ocular melanoma patient who I actually took care of, and you can see he's got a nice fluffy hepatic metastases there that became attenuated, which usually indicates necrosis, and then it shrunk, which was nice. We like to see that. And this was a patient with an ovarian cancer at Georgetown who had a solid tumor in her peritoneum that shrunk very nicely, 75% decrease, and a patient with renal cell carcinoma. I can't remember where this patient was. It might have been here. Here, yeah, it was site 01, who had a cardiac nodule in the pericardium there that went away completely, a lung nodule that shrunk significantly, and a diaphragmatic nodule that shrunk significantly. So we saw some signs of response, and that was very encouraging. We want to move it forward, get into a phase 2 trial and all this. Along the way, we were collecting PBMCs for biomarkers to measure F2,6-bisphosphate, the product of this enzyme that we were targeting. So we decided to look and see what was going on with the immune system. And what we saw, this is just a single patient. This is a patient with stable disease of breast cancer. We saw all the suppressor cells go down when we gave the agent. And then we saw all the T cells, the CD4 and CD8s, get hyperactivated when we gave the agent. So it really fit with our model, which we were excited about. But even more interesting was when we looked at, we only had 4 patients worth of serial peripheral blood monocytes. But what we saw was that the patients who had some clinical benefit from the agent were the ones who had the biggest decrease in Th17 cells, the suppressor cells, and the biggest increase in the activated CD8 positive T cells. So that was very promising for us. And so we're kind of starting to work on this and going back to the lab and trying to figure out how to combine this agent with other immunotherapies. And this is our model system, but the first step was to try it with anti-CTLA-4, and we did that in that B16 model. And what we saw was that when you combine it with an anti-CTLA-4 in a model that, I can tell you that if I did the same model with anti-PD-1, the shift would look like this. You can see how ineffective our preclinical models are at predicting clinical success in humans. But we did see a nice shift when we combine it with anti-CTLA-4. This was done by Sucheta Tailan. And so the third and last cause of immune resistance is collective term T-cell anergy. It can be from a variety of reasons, but it happens, and we know we need to come up with ways to superactivate the immune system. And one way to do that has been studied at the National Cancer Institute for about 20 years now is called tumor-infiltrating lymphocytes. So basically what you do is you take the tumor out, you isolate the infiltrating lymphocytes. These are all terminally differentiated CD8 positive T cells. They used to isolate them by PD-1 expression. So they were called exhausted because they didn't respond. And then what you do is you give the preconditioning chemotherapy. It's fludarabine and cyclophosphamide. So you drop the patient's white count down to 0 in the bone marrow transplant unit, and then you infuse about 100 billion of these tumor-infiltrating lymphocytes and give one cycle of a growth factor for the lymphocytes called high-dose interleukin-2, just one cycle. And so we're doing this. So it was done at the NCI and then at MD Anderson. They picked it up. I tried to set it up here. It costs about $2 million a year to run a program like this. It's not FDA. It's not, you can't get insurance coverage. But then a multicentre trial became available. It's 7 centers in the country. We're the only one that's not on a coast. We're the only one in a state that's not on a coast. So we get patients from Denver now, and from Tennessee, obviously. And so you can't go. The closest place for most of the patients in the country is to come to Kentucky. That's why UPS is here. And this is one of the patients. He had stage 4 disease. He had been told to go home and die. He'd gotten his BRAF inhibitors. He'd gotten his immune checkpoint inhibitors. He'd gotten everything you can imagine. There was nothing left to do. You can see from his prior. I'm just showing one of his many tumors, but you can see this large lung mass. And then he went on TILs in July, and he responded very nicely. He's alive and well, taking care of his kids and his wife now. And so we're really proud of that fact. And we're now opening up the phase 2 trials for lung cancer and head and neck here, and probably going to go to cervical cancer after that. And we're a super center for this particular trial. This is a poster that we presented. The whole group did. It's all 7 centers. But this is in melanoma. These are all patients who were refractory to the immune checkpoint inhibitors, and you can see the majority had their tumors shrink. They didn't reach the criteria that we used. RECIST criteria is 30% of the maximum diameters. Based on the index lesions, but we still had an objective response rate of 29% in a highly refractory population. And then the last T-cell anergy example that I don't have data on, but I just have to mention, is William C., who's the head of our bone marrow transplant division, is in a close collaboration with someone who's from a similar region in China, UofL Ma. He's now at Stony Brook. And you've probably heard in the news about CAR T cells, chimeric antigen receptor T cells. All that is, it's really quite simple. It's just hooking up the stimulation, the signaling domain of the T cell receptor with an antibody against a certain antigen. That's all it is. It's not rocket science. We could have done it in the lab 20 years ago. But Carl June had the courage at University of Pennsylvania had the courage to put it into kids. So what they do is they take T cells out, autologous T cells, and they transduce the T cells with a virus that packages this fusion protein between the antibody and the T cell stimulatory domain. And then they put the cells back into the patient. And B-cell leukemias in kids who had failed or progressed through multiple other all available regimens, they had an 83% response rate. These kids were all going to die. and 83% aren't dying now. I don't know, it might be 70%, but they have an 83% complete response rate. So that just got FDA approved, but 3 weeks ago or 4 weeks ago, through Novartis is now took on the technology and is charging $500,000. So these two are working together to use it, and it turns out Dr. Ma apparently was the mentor of Carl June, who developed the technology that just got FDA approved. Dr. Ma is trying to do this for other cancers, the more difficult ones. And the two big ones that they're interested in are T-cell lymphomas that are refractory to standard therapies and multiple myeloma, which combined add up to about 25,000 deaths per year. And this is a great example. So all these are fluorescently labeled T-cell lymphoma cells in an animal, and they all concentrate in the bone marrow of the mouse. And you can see what happens over the course of just a couple weeks when you inject these cells. The animal subsequently dies. Well, if you give a CAR T-cell that's targeting the CD4 receptor, which is T-cell receptor, you can completely eradicate those cells. But what do you think happens? If we did this in humans, you'd end up with AIDS, right? If you did this. So they've come up with some pretty smart approaches to dealing with that potential. One is to use natural killer cells that are more short-lived. Another is to use a suicide gene approach. And the third is to use drugs to get rid of your CAR T cells. But again, these are patients who are really headed to hospice. And I think it's going to be, I think we're going to see some exciting results out of these trials. There's two that are opening here at the Brown Cancer Center. One is for refractory multiple myeloma, and they call it a compound CAR, never been developed before into clinical trials, but basically you target two antigens at the same time. And then for refractory peripheral T-cell lymphomas, with the CD4 CAR, they just got orphan status for that. And so this is my last slide. I believe this. Now, is it going to be 5 years or 10 years? I don't know. It might be 5 years. I believe that immune checkpoint inhibitors are going to drop the United States cancer death rate by 25%. I've done a little bit of the math with melanoma and non-small cell lung cancer, renal cell carcinoma, head and neck, and there are assumptions that I'm making, some of which include our ability to combine these agents with other agents. But I think we're going to drop from about 600. Right now, the estimates are that the cancer death number in the United States will go up because of the baby boomers. It's going to go from 600 to 650 or something over the next 10 years. Globally, it's going to be much worse than that. It's going to go from 8 million to just under 20 million. But I believe it's going to go down. I think it's going to go down. We're going to hit like 400 probably in the next 5 to 10 years because of one class of drugs. That's how incredible this is. I think that we need to invest a lot of our resources in trying to figure out how to overcome intrinsic and acquired resistance to these agents, and certainly that cell-based therapies hold great promise. And I'm going to stop there at 9 o'clock sharp. Thank you very much for holding back.
Very exciting. I had a couple of questions.
Yeah.
One is aging.
Yeah.
Do these PD-1 and other checkpoint molecules increasing with age?
So what happens is if you look at T cells in the elderly population, you will see higher levels of CTLA-4 in those patients. And in fact, now it's never been proven, but we've done an analysis that we still haven't published where we looked at the response rate of patients who are over 60 to anti-CTLA-4 versus patients who are under under 60. And the ones who are over 60 respond at a higher level. Which, to me, the basic idea is that if you've got a 30-year-old who's got cancer, it's probably because it's a pretty damn smart cancer. It's got a lot of mutations, it's driven, it's doing its thing. If you've got a 90-year-old with cancer, it's probably because their immune system is faltering. It's coming down. So that made sense to me. Those older patients, and we see that all the time. The older patients tend to do better.
Okay.
Yeah.
Yes, sir.
Yes. Yes. Not here, but there are open. It was an idea that we had that we talked with Nejat Egelmitz about, and we put into he's got an internal grant but unfortunately there are bigger institutions with a lot more funding that could move more quickly through that idea. But the answer to your question is yes, there are the microbiota does predict who's going to respond, but more so who's going to have the toxicity, in particular autoimmune colitis from it. And certainly changing the flora would, and there is one trial open right now, we don't have it open, where they're trying to affect the flora and see if they can get better response rates. So it's a good point.
What percentage of tumors express this PD-L1? And from your talk, it's my understanding that apparently the agents PD-1 inhibitors work in tumors that don't even express PD-L1.
That's right.
And what is the mechanism there?
Yeah, so PD-L1 is expressed by antigen-presenting cells. So antigen-presenting cells, by that I mean macrophages, dendritic cells, Langerhans cells, you name it, all express B7-1 that engages CTLA-4 as well as PD-L1, and it turns them off. So that's the mechanism. Now, in certain cancer types, in particular non-small cell lung cancer, there's a very strict correlation between PD-L1 expression in the cancer cell versus the antigen-presenting cell. In others, there's no strict correlation. There's a small advantage if you have PD-L1 in the tumor. So melanoma, for example, doesn't exist. And that's presumed to be all central lymphatic responses that are occurring with the antigen-presenting cells in the lymph node, interacting with the CD8-positive T cells. Yeah.
Questions, comments? Okay, so one last question for you. You mentioned that the animal models are really a poor representation of humans.
Yes.
Of course. But you still need preclinical data for some of these. Is it mainly to evaluate safety as opposed to effectiveness?
Well, yeah. I mean, you want to look at both those questions, obviously, but where the money is. So if you bought into my approach, which is if we're going to cure cancer, I know if we're going to cure cancer, we're going to do it through the immune system. If you believe that, then what you want to do is PDX models, so it's patient-derived xenograft models, and what you do is you take an animal who is immunocompromised, you put the patient's own tumor into that animal, you put the patient's own hematopoietic system into that animal, you reconstitute the immune system, and now you have a humanized PDX model, and that's where you do your preclinical experiments for efficacy and toxicity. I think toxicity is pretty much handled. I'm not really that worried about that as much as it's the efficacy. We've not seen a lot of toxicity disconnects, let's say, but the efficacy, clear disconnect. The mouse immune system is very different from the human immune system. The models are horrible for the most part. Either they're transgenics that are driven by an oncogene and a loss of a tumor suppressor, basically 2 mutations, unlike 10,000 in melanoma, or it's going to be a cultured cell line times 10 years that's been put into a syngeneic animal. So these PDX models are really quite compelling. We've developed a program here where we're partnering with a company called Champions to provide bone marrow biopsies from leukemic patients, solid tumor tissues, and we're creating a library, it's a repository of PDX models. It's all in Houston. And with the next step being, and this is the hard part, we've got to convince our patients to get leukapheresis, to get those immune cells back into those animals. And that's where we're headed with all this.
Interesting. Well, in 2025, we will bring there will be a chart on the death rate of cancer.
Thank you all.
And we'll calculate whether it's down at 25%. But based on these data, we're excited.
Thanks a lot.
Very good. Thank you. Thank you. Appreciate it. Jason's going to take a picture.
Oh, okay. Hey, good to see you. Yeah.